Hi, hello.
Hello, everyone. Welcome and thank you very much for joining to this invited session titled Women in Biostatistics, Insights from the BioStatNet Network. It's a pleasure for me to present our four speakers today: Natalia Vilor Tejedor, Naomi Diz Rosales, Alba Muñoz, and Silvia Pineda San Juan. And also I would like to thank Jaione Etxeberria for organizing this session. Jaione will start presenting briefly the BioStatNet network. So Jaione, whenever you want, the floor is yours.
So thank you very much, Irantzu. This is a pleasure for me to present or to be part of this session, Women in Biostatistics, Insight from the BioStatNet network. And before starting with the speakers, I would like to first thank the organizers for organizing such a lovely event and accepting the BioStatNet session to be part of it. And before we begin, let me take a couple of minutes to briefly introduce our network we are representing today, that is BioStatNet. So, established in 2010, BioStatNet is a national network that connects biostatistics researchers across Spain. And it serves as a collaborative platform to first enhance proper training in biostatistics, coordinate research and teaching in biostatistics within Spain, enabling its international projection, and also to promote the transfer and application of biostatistics in biomedicine. So currently, we are more than 320 members, including statisticians, computer scientists, and biomedicine experts, organized into 12 regional nodes across Spain. And we are indeed a very active network because having already celebrated 5 general network meetings alongside 5 member assemblies, we hold periodic executive committee meetings for coordination. We also promote in-person scientific stays for product students, promoting scientific synergies among the youngest network members. And we also regularly introduce or organize different scientific workshops, such as BioStatNet software workshop, Susi Bayarri Women and Biostatistic workshop, BioStatNet workshop on biomedical big data, etc., etc. So to know more about us, please scan the QR code you have here. And having said that, we have 4 women representing the network, as Irantzu very well says. They are Natalia Vilor Tejedor, Naomi Diz Rosales, Alba Muñoz, and Silvia Pineda San Juan. So thanks all for being here today. And please, Natalia, if you wanted to share your screen.
Could you hear me well?
Yes.
Sure.
Yes, okay, I'm gonna share my screen. I think you can now see my slides.
Yes.
Yeah. Great. So yeah, thank you so much, Irantzu, Jaione, and also the BioStatNet and the organizers of this International Day of Women in Statistics and Data Science. To me, it's a pleasure to be here as a representative of BioStatNet, showing a bit my stochastic journey, I call it like this, but also the journey of my team that is actually, they are the one responsible for me being here today presenting what we are doing. And yeah, well, Jaione was mentioning a bit about the connection between biomedical research and also mathematics and statistics. And this is actually my background. My background is in mathematics, but I moved during these years through this nice journey of biomedicine to try to apply all our mathematical knowledge into this biomedical background. And actually, my interest is focused on trying to understand and apply these methods to understand the differences between us. What are the things that make us different? Because this is very related with our vulnerability to several diseases and also to several biological processes. And although in our team we are working on several aspects, first of all, to try to understand how these risk factors or these differences contribute to our health and disease, today I wanted to spend these minutes talking about the statistical modeling or the statistical methods that we are developing or using in our group, but also I want to keep a bit of time for this part of inclusiveness in science. I think it's something very relevant that we should also start to or include in our modeling and in our methods. So to put in context a bit, we are working on the field of neurodegeneration, and especially in the context of integrating genetics and brain health, or omics and brain health. And our objective or main goal is to try to understand which genetic factors, variants are associated at the end with a neurodegenerative process. The problem here is, what we find is that these neurodegenerative processes are sometimes variables or status that are defined by a clinician, and that are normally categorized between cases and controls. But this is tricky and so heterogeneous, especially for neurodegenerative diseases, and this leads to a lot of misclassification. So in this context of the neurodegeneration, what we try to use is some intermediate proxies that are our brain, basically, or our brain features, to try to discern first the genetic variants associated to these outcomes, to these brain features, and then try to infer how these changes in the brain are gonna affect our disease status, more in a continuum way, but not in a dichotomous way. And to do that, what we normally do is something called GWAS analysis, wide association studies, that statistically we go through the statistics. Basically, the bases are univariate linear models in which I test each independent genetic variant against my outcome of interest, which is going to be one of these brain regions. So this is the gold standard that was used several times. But the thing is that at some point, or we increase our sample because we have a lot of different variants, millions and billions of genetic variants, and also different brain regions and points in our brain. Or if we don't increase this sample, we have a problem. We have a statistical problem, power, but also a biological problem. On that sense, what we started to do, or what I started to do in my PhD, and that we are things that are continuing now in the team, is something called methodologies called gene set analysis and also polygenicity. So basically, what we are going to do in this type of biological concepts is a multivariate methodology. We're going to try to group the genetic variant, the individual ones, in a more representative biological composite, which is the gene in which they are included, or which is more a score that is summarizing the genetic risk through a specific disease or trait. This is something that we are trying to improve because at the end, as I'm saying, we can make this composite based on the biological meaning, the gene, but there's other things like functionalities and things that are not taken into account. Most of the methods are focused only in p-values, and I think we all know that this is perhaps not the best way to do it. So we are trying to find a way to clusterize them in a more relevant way. The other thing is work that is being done by one of my PhD students, Patricia, which is trying to understand also our brain as a complex system and as a multivariate thing, not just as a univariate region. Because we all know that if there's changes in one region, there's going to be probably other changes in other places. There's a kind of a covariance or dependence between the brain regions. So for that, we really need to also try to understand and to develop tools that allow us to quantify this and to quantify then the genetics behind these multivariate changes. So these are things that we are developing. Other things that I find very interesting are also the part of improving our knowledge on neurodegeneration and healthy aging, taking or using other type of omics like proteomics or transcriptomics. And this is on one side very useful and biologically useful, but on the other side, the problem is that we really need post-mortem samples to assess this information, because we need to go through the brain, and you know, it's not easy to get all of this information. So there's some tools that we're trying to develop as well, which are proxies of this information in order to try to address the challenges of getting this data. Other type of things that I think are very interesting for us is when we are trying to model some outcomes that are not following normal distributions. So I didn't mention that, but normally in GWAS studies, we use, in our regression models, we use logistic regressions or linear regression models, assuming all the assumptions behind. But it's true that for some features, for instance, for cerebrovascular features, the normal distribution is not there. And there's a lot of assumptions that we cannot assume. So we are also trying to develop other non-parametric methods to try to address this association between genetics and these brain phenotypes that can be actually extended to other types of biological information that is not following these normal distributions. A third aspect that we are evaluating in the team is the integration not only of genetics, but also of the exposome, the environment. And this is also very tricky. It requires extensive mathematical modelization, not only in the last step of combining the data, but also in the design of the projects and of the experiments. It's very relevant to take into consideration all of these factors, because actually at the end, it's not 100% genetics, it's not 100% the environment. There's a combination between different aspects that we really need to take into account in our models to then address the influences on our brain, on our health. And the last part that we are also exploring is, of course, the use of different machine learning and artificial intelligence methods, especially for disease subtyping and stage classification. Here, we are now working with some autoencoder models. We were previously working with some classification models to try to know if using this brain data, we are able to classify, for instance, Alzheimer's disease patients versus controls, or from different types of dementias. And I think this is very promising, especially now that all of these tools are getting, let's say, famous and more sophisticated. But actually, something that I wanted to mention is the inclusivity in science, the inclusivity that we also need to include in our statistics, mathematics, because otherwise we are not doing research for everyone. And this is a clear, well, this paper is very nice, I like it a lot, and it shows very clearly why we are still not at the right place in science. And this is an example of studies, genetic studies. And here you have in different colors the different populations that are used in each of these studies for discovery and for replication. And as you can see, almost all the studies are blue, which means that they were using European populations. This has several implications. Especially for then the applicability of the results that we are trying to develop. And not only for genetics, also for environmental risk factors, modifiable risk factors. This is also a very nice study that I recommend you to read that was performed in New Zealand. And they were evaluating the impact of different risk factors related to dementia in 4 different populations living there in New Zealand. And for instance, if you take a look on air pollution, for instance, here these are the odds ratios. And here for the Pacific people, for instance, it's 2.5, but for Maori it's 1.8. So here it's 2.6. So we see that even for people that are living in the same place, the ethnicity is making a difference on the impact, not the vulnerability of these risk factors. So all of this needs to be taken into account in our models. And this is why we actually created this consortia 6 years ago, in which I'm the responsible of the statistical modeling, the United Consortium. We are trying to really care about the inclusivity in neurodegeneration and in general in brain health. And we're developing also things to do that. For instance, evaluating different factors that are affecting this diversity between populations, this brain health diversity, through analyzing different parameters that we take into account when we acquire these brain images and brain measurements. Also, we are developing some tools, deep learning tools, to try to reconstruct measurements from brain scans that were obtained through CT instead of MRI, which are tools that are more accessible in some places in South America, for instance, and not in like MRI in Europe. We are also working on trying to discern the role of the local ancestry, local genetic ancestry, because in most of the studies, we focus on our self-reported race or even in a global genetic ancestry that is computed through a PCA. But we see, and there's examples like this paper, that even individuals that self-report the same and have more or less through PCA the same percentage of one ancestry, when you go locally to different chromosomes, you see differences. You see here more red and blue than here. So there's here a mixture of European, African that is not here. And we are only taking into account this in our model. So I think this is also something very relevant as well to take into consideration. And finally, of course, now with all these machine learning tools, we really need to be careful with the prompts or the information that we are introducing to them. And for this, we were grateful to receive and to be partners of these two European projects that they are going to try on one side to evaluate some aspects of inclusivity in biomedical research centers, and also another one that is going to try to evaluate also these bias and EI tools. And with that, I wanted to just finish. This is the roadmap. I don't have time to go into other aspects that I find very relevant, but I wanted to just say that to me, actually, one of the important parts that we really need to consider here is this clinical translation and social health. And the end, on my personal level, if I'm doing this research and trying to apply all these computational, mathematical, statistical, biomedical things, is actually to have a better society and better things for being applied in clinics. So yeah, just to thank my team that, as I said, are the ones that are doing this amazing work, and also all the partners. And again, thanks to BioStatNet for this time and the organizers of this really great initiative. Thank you so much.
Thank you very much, Natalia. That was a really nice presentation. I didn't get any message from the chat, but I think Jaione has at least one question to ask you. If anyone else has something that wants to ask to Natalia, please write it in the chat and I will let you know.
Yes, Natalia, I have a question related to a couple of things. When you have shown us the slide that was entitled Assessing Diversity from Multiple Layers of the Brain, in that, for that specific study, you use some kind of spatial modeling or something like that? Or
you mean the first study that I was showing?
No,
the
I think it was one of the last one. One of the latest. I think the slide was the.
In that part, I showed 3 studies mainly. The first one, we evaluate different MRI acquisition, like brain image acquisitions, taking into account different parameters of the brain scans and also the different populations and ethnicities. And what we found is basically that the parameters of the scanner and all these more technical tools are not as important, and actually the population that you are scanning. This was the first study. Not sure if this was the one that you were mentioning. The second one is very interesting. I didn't put a lot in the context, but because I didn't have time. But here what we are doing is to try to apply some deep machine learning and deep learning tools to try to reconstruct low field MRI and let's say low resolution images. Why? Because what we discovered when we were traveling in South America is that when you go to the hospitals, they don't have this big MRI and the magnetic resonance imaging machines that we have here. Like we have huge tools. There, what they have are the CTs or a low field MRI. The problem of this is that the resolution is not sometimes useful for anything, not for research, of course, for clinical practice, just a bit. So what we were trying to do is to apply these machine learning methods to develop them, to try to reconstruct these low resolution images into a high quality image. And then with this, we are seeing that actually the agreement between the high quality one and the one obtained through the magnetic with the big machine are actually in agreement. And this is helping a lot because a low field MRI is a 10-kilogram machine that you can move in a van. So in Peru, for instance, that they only have one MRI in Lima, one MRI in Cusco, you can drive and go to a city town and scan people. And this is very, very relevant, of course, for a medical practice, but also it allows for us to have information and to provide a tool to the medical doctors to evaluate these patients there, not just, having a machine that is getting the information. And the third one was more genetic related. I'm not sure if it's the one that you were mentioning.
Okay. Thank you, Natalia. Very, very interesting, but I think we should move for the next speaker. Thank you very much for your great presentation.
Thank you.
Thank you, Natalia. So now we'll go for the next presentation given by Naomi.
Can you hear me?
Yes.
Yes, sure.
Could you see my slides?
Yes.
Perfect. So, hi everyone. First of all, thank you so much for the invitation to stay in this fantastic Congress. I'm Naomi, I'm part of the BioStatNet Galician node, and I am a biologist who has embarked in the adventure of doing a PhD in statistics because I'm crazy. And currently I'm at Universidade da Coruña in Galicia, and I'm working under the supervision of the four professors, María José Lombardía from the Universidade da Coruña and Domingo Morales from Universidad Miguel Hernández de Elche. And today I'm going to talk a little bit about a small area estimation for social and biomedical problems. So, what will be our roadmap today? I will start by introducing our research line. Next, I will move on to understand our main objective. I will define our methodology, but I'm going to focus more on the application to real data. And finally, at the very end of the presentation, I will provide you with some conclusions and key reference, just in case you were curious about our work after these 12 minutes, I hope so. So let's start. Well, we can start by introducing our research line. So we are trying to develop and apply random slope mixed models to estimate complex indicators in small areas with two main objectives: the estimation of occupancy by COVID-19 or, for example, by flu in intensive care units, and the estimation of the poverty ratio. In this presentation, I will focus more on the first objective, but at the end of the presentation, I will give you some highlights about how to apply our models into other different contexts, that is the socioeconomic context. So we are trying to develop and apply random slope mixed models for small area estimation. So we have to understand what is small area estimation. Let's step by step. Well, small area estimation is defined as a multidisciplinary branch of statistics with this objective. I have an area or domain. For example, it could be a geographical domain or a social-demographic domain. And I want to estimate an indicator of interest, for example, the intensive care unit occupancy by a disease. But I don't have enough data to do it in a precise way. So I have a problem, and there are a lot of techniques available in small area estimation, but we are trying to focus our work on used mixed models. A mixed model, like this model that you can see in the screen, is a regression model that combines fixed effects with random effects. And why is this interesting in small area estimation? So I have here my favorite frame where you can see the Galicia map. It's an autonomous community in Spain. And here we can see the 7 health areas in Galicia. Well, just imagine that I want to estimate an indicator, for example, the intensive care unit occupancy in this area today, but I don't have enough data to do it in a precise way. So I can put the same random effect here and here, similar areas, and in this way, I increase the effective sample size and the precision of my estimates. So mixed models, because unity is a strength, and in this way we can improve our metrics. There are a lot of examples in the literature with this kind of models. It's a linear mixed model with a random intercept. That is to say, for example, I suppose that each health area has a different starting point on occupancy. But we ask ourselves, is this always enough to model heterogeneity between our areas? Here, I represent 2 examples. On the left, we can see the relationship between our target variable, the intensive care unit occupancy, versus auxiliary variables like variables that measures the severity of the disease for each of the 7 health areas in Galicia. And we can see that depending on the health area, the slope is different. The relationship between our target variable and our auxiliary variable is different. Here on the right, we can see another example in the socioeconomic context. For example, I want to explain the poverty ratio as a function of auxiliary variables like the unemployment rate. And I can represent by province. Each dot is a province. And here we have 5 lines because we have grouped the provinces into income groups. We can see that depending on the income group of the province, the relationship between my target parameter and my auxiliary variable is different. So we have different slopes. So we ask ourselves, why not incorporate random slope to mixed models for estimate in small areas? And we searched a lot and we only found that in 1981, it was the first job about random slope mixed models with a small area estimation in a context of a linear mixed model. And the results were better both in simulations and in application to real data. But since then, there are few literature available, it's computing demanding, etc. And for example, there are no works about generalized linear mixed models, very useful for count variables, with random slopes and random intercepts in a small area estimation. So what a surprise, our objective was precisely to develop and apply random slope generalized linear mixed model to estimate in small areas. And remember that we have 2 objectives. So let's go with a little bit of our methodology. I will explain briefly our model. We have called the model the area level random regression coefficient Poisson model. Just a note that random regression coefficient is the same that random slope. And here is the name, ARCP model is for the abbreviature of our long name model. Here we have the reference where this work is published. And at the end of the presentation, I will provide with the complete reference, just in case you were curious. And I want to share a little bit of notation. So here we can see our target variable denoted as Yij, that represents, for example, the number of patients with COVID-19 in intensive care units in the health area I and in the JJ. We have a total of D domains, the cross between the health areas and each of the days. And we want to estimate a target parameter that we have denoted as Pij, that is the proportion of people with COVID-19 in each health area and day. Well, we have our target parameter. So to explain this target parameter, we assume that we have explanatory variables. And here we define the random effects. We define a random intercept, a different starting point, depending on the health area and the day that we have denoted as Uij, that follows a normal 0, 1. And we have defined for the first time in a smaller estimation, a vector Vk of random slopes that follows a multivariate normal with this dimension P, because it depends on the number of auxiliary variables who has the random slope. Here the mean is a vector of zeros, and here you can see a strange symbol that is the variance covariance matrix. I present you in this frame, where on the diagonal we have the variances parameters, always greater than 0, and both sides on the diagonal we have the covariances, where rho represents the correlations between the random slopes, and it's always between -1 and 1. So we have our target parameter, we have our auxiliary variables, and we have our random effects. So we have that our target variable, Yij, conditional on these random effects, follows a Poisson distribution with this mean, where nu ij is a sample size of the offset. It's always known and greater than 0. And remember that Pij is our target parameter, the proportion of people with COVID-19 by health area and day. Well, this is the final relationship of the natural parameters of the model, where here log is the link function because we are dealing with a Poisson distribution. Here we have our offset, our fixed effects, our random intercept, and our random slopes. And we only have to estimate this vector zeta of unknown parameters, beta regression parameters for the fixed effects, and sigma, phi, and rho are the variance and correlation parameters for the random effects. We also have to predict the random effects, the values for the random intercepts, and the values for the random slopes. And we are doing so by a maximum likelihood Laplace approximation algorithm using the free statistical software R. So once we have defined our model, we define the best predictors under this model, and it's not enough to get a point estimation. We also have to provide with an error measure. So we have defined the mean square error estimators, and we have conducted several simulation studies mimicking the COVID data, mimicking the poverty data and the socioeconomic problem, and we have selected the best predictor and the best mean square error estimator in each case. Now I'm going to focus on application to real data, but I can answer whatever question about that. So let's go with application to real data, and just remember that we have two objectives. Apply this random slope generalized linear mixed model to estimate intensive care unit occupancy and the poverty ratio. I'm going to focus on the first one. So our target variable is ICU, the number of patients with COVID-19 in intensive care units in the health area I and in the day J. To explain this target variable, we have these auxiliary variables that measures the level of transmission of the virus, the level of utilization of care services, and the level of severity of the disease. The domains are the 11 health areas in Castilla y León and the 7 health areas in Galicia, because Castilla y León and Galicia, that are two autonomous communities in Spain, were the most affected and the least affected by the COVID-19. And we have studied these health areas from the 2nd November of 2020 to the 6th of March of 2022, because we want to consider different virus variants, different epidemic periods, different authorities measures, and check the flexibility of our model. We have constructed the database from scratch, and we have made it available in a GitHub repository. I provided here the link. And let's see the results. We have checked the estimation capability of our model and the prediction capability of our model. So we have fit our model from the 2nd November 2020 to the 2nd of May 2021. And here we can see in black the observed intensive care unit occupancy, in red the estimation under our model, and in purple the root mean square error. And we can see that the results are good. But it is one thing to estimate well, and another different thing is to predict well. So we have evaluated the prediction capability using the technique rolling origin forecast. It's easy to understand. I fit my model from the 2nd November of 2020 to the 2nd of May of 2021, and I predict from the 3rd of May to the 9th of May. In the next iteration, I fit my model from the 3rd of May for the 3rd of November of 2020 to the 3rd of May of 2021, and I predict from the 4th of May to the 10th of May. And so on and so forth. I have evaluated the capability prediction of my model more than 300 times. And here are some of the results. Again, we have two health areas. I represent in black the observed occupancy, in red the prediction under our model, and in purple the root mean square error. The error is greater than the estimation scenario, like it's natural because there are more uncertainty in the future, but the results are good enough for health authorities, and we can provide them with this kind of risk maps. This is Castilla y León, and the higher the color intensity, the higher the risk of collapse of intensive care units. And we can provide these maps 1 day, 2 days, 3 days, until 7 days ahead. So it's really useful to manage the hospital resources. All this methodology and data description is available in this archive paper. Well, and I'm going to finish providing some examples of how to apply this model to estimation of the poverty ratio. We have applied the same model, to poverty data in Spain to estimate the poverty by province and sex. And here the Spain map, the poverty map, where the higher the color intensity, the higher the poverty risk. And we can see patterns increasing the poverty from the north to the south and the east to the west. And we have detected significant difference between women and men, with more poverty in women than in men. So this kind of maps provides with very valuable information to the authorities. And here is available in open access our research. So let's end. We have defined for the first time in a smaller estimation this kind of model, a generalized linear mixed model with random intercepts and random slopes. We have applied it to estimate and predict occupancy of intensive care units, and we can provide with this kind of risk maps. We have applied the same model to a socioeconomic context, and we can provide the authorities with this kind of poverty maps. And right now we are working to increase the flexibility of our model by defining time correlations between the random intercepts. So let's see what happens. Finally, I would like to thank BioStatNet and the funding received. Here are some of the references that I mentioned in the presentation. And I want to end with this picture. It's the Torre de Hércules, a World Heritage Monument in my city, A Coruña. And I like to think that in this Congress, we act like a lighthouse, shining a light on the way for women in data science and help reaching new destinations. So thanks a lot for inviting me.
Thank you very much, Naomi. That was a really nice last picture to end the presentation. I'm sorry, but because of the lack of time that we have, I think we will leave all the questions for the end of the session, if that is okay with you. And so we will move to the next speaker. And if anyone among the attendees wants to ask anything to either Naomi or any of the other speakers, you can just write it in the chat, and we can comment on that at the end of the session. So we can move to Alba Muñoz.
Thank you so much. I'm going to share my slides. Yeah, I think you can see my slides now.
Yes.
Okay, thank you so much. So yeah, my name is Alba Muñoz del Río. Now I'm working at Universidad Distancia de Madrid, but this research has been carried out with my two supervisors, Víctor Casero Alonso and Mariano Amosalas, of the group Optimal Experimental Design in Universidad de Castilla-La Mancha. So I would like to begin this presentation with these two news. I'm sure these types of headlines sound familiar to you because food poisoning outbreaks appear in the news quite often. And behind each story, there's a very complex question, which is under what conditions do microorganisms grow, survive, or die in our food? So understanding that is what predictive microbiology is all about. Predictive microbiology is the field that develops and applies mathematical models to describe how microorganisms behave, how fast they grow, or how they respond to ambiental factors like temperature or pH. With these mathematical models, we can predict how long food will remain safe or under which conditions bacteria will stop growing. So these models describe microbial responses in two forms or in two layers. First, we have the primary models, which capture how microbial population changes over time. For example, how quickly it grows. And then we have the secondary models, which describe how the kinetic parameters of the primary models, like, for example, the growth rate, vary with factors such as these ambiental factors, such as temperatures. In order for these models to provide reliable predictions of microbial behavior, experimental data are required to accurately estimate their parameters. So here is where optimal experimental design enters the scene, because collecting those data can be very resource intensive. For every microorganism and every type of product, we need to test several conditions, like temperature or different points of time. And we also will need to test with replicates. So not all experiments contribute equally to improving our parameter estimates. Some are very informative, while others are add little new information. So the challenge here is how can we plan experiments in a way that maximizes what we learn from each one. So here is where the design of experiments becomes crucial, because optimal experimental design uses statistical tools and criteria to identify which combination of conditions will give us the most precise estimates of our model parameters. So in other words, it will tell us where it's worth experimenting and where it's not. So I don't have the time to get too technical, so I won't explain the statistics behind OED. Instead, I'm going straight to our case. So our goal here is to examine or analyze the standard parameter estimation procedure used in predictive microbiology and show how optimal experimental design can optimize it. We are studying a real case study which measures the growth of Pseudomonas on button mushrooms, carried out by Tarlac et al. At 2020. And this study is using the primary model, which is called Barrio, which describes the growth of these microorganisms a long time. So then we are going to be using the Rutkowski square root secondary model, which relates the maximum specific growth rate with the temperature. So in other words, we want to study how temperature influences this growth. So the traditional procedure will begin with several isothermal experiments at different temperatures. As you can see, we have 4 different experiments at 4 different temperatures. And in this case, the authors propose these 4 experiments at these 4 different temperatures. So as you can see, they propose having or testing or measuring this microbial population at these different hours. Okay, so here, using optimal experimental design, our work provides the corresponding optimal designs for each temperature also. These designs, optimal, guarantee maximal joint precision in estimating all model parameters. So as you can see, here we have the designs. I want to explain what is this. The first row listed for each design contains the observation times, and the second one contains the proportion of observations of the total experiment that should be carried out at each observation point. So in our case, as you can see, we only need 4 observation times compared with the 7 or even 8 that the authors needed. And moreover, if we look at this last column, this one reports the efficiency of the author's designs and the relative to our optimal designs. As you can see, this efficiency indicates that by taking only approximately the 80% of observations in our optimal designs, we would match with these ones, and that 80% of observations, we would match the estimation precision that the authors obtained with these designs. So in practice, this means that our designs require fewer observations, which together with the reduction in observation points translates into lower experimental costs. Then in the usual predictive microbiology workflow they follow, based on the experiments conducted by the authors, the parameters of the primary model are estimated. And among these, it is the mu max parameter, which is the one that is precisely related with the temperature through the secondary model. So in this way, the authors, as you can see, obtain an estimate for this parameter at each temperature. So for them, they use these 4 points, these 4 combination of temperature and estimation of the parameter, and fit this secondary model, which statistically speaking is not the best way to do it. But also this procedure leads to ignoring the uncertainty associated with the estimation of mu mass, which statistically speaking, again, is a serious issue. In the second stage fit of the secondary model, that uncertainty is overlooked, and therefore the new estimates are less reliable. So for this reason, using optimal experimental design, we propose employing the C-optimal designs, which ensures maximum precision or minimum variance in the estimation of a single parameter. So in this case, we propose specifically calculating the C-optimal designs for mu max, which is the parameter that we are interested in. So this is, we are going to estimate this parameter as precisely as possible, and when fitting the secondary model, the associated uncertainty is minimized. So as you can see now, the C efficiency of the author's design, which is this, this column here, doesn't exceed the 50%. So as I have said before, this means that with half the number of observations in our designs, we would match the same precision in the estimation of this parameter as the authors. Moreover, if we take a look at this one last column, we can we calculate the efficiency of our previously proposed optimal designs, and these ones exceed 80%. So this means that they deliver a more precise estimate of mu max than the author's original designs. Now, if we truly want to apply an optimal technique and we don't want to just follow this traditional workflow or this traditional procedure, we can optimize time and temperature simultaneously by embedding this secondary model, which is the function of time, into the primary model, which is the function, which is the function of time. The secondary model was the function of temperature. So then we can compute the optimal design that maximizes the optimal designs, I remember, optimizes the joint precision in estimating all the parameters, both of the primary model and the secondary model, which is really our final goal. So here we obtain a design that tells us when to sample over time and also at which temperatures. As you can see, this design leaves the temperature of the first observation and the last observation to the experimenter's choice. And it establishes this relationship between the temperature and the experiment's final time. So, as I mentioned, I can go too deep into the technical aspects of the design, but this result lets us optimize not only the observation times, but also the temperatures, and it defines a function for the final experimental time so that the experimenter can decide whether to prioritize time cost or temperature related cost. So as conclusions, we have evaluated this standard procedure for estimating the parameters of primary and secondary models, and then we have applied optimal experimental design techniques to optimize it. So per temperature, the optimal designs have achieved higher joint precision with this between 15 and 20% loss of efficiency of the author's designs against ours. And then for precise secondary fitting, estimating mu max, this parameter that we are interested in. We have calculated these optimal designs, and the authors, these proposed designs obtain C efficiencies lower than 0.5%. Also, we recall that our designs, our optimal designs exceed this 80% of efficiency, and in the real optimal solution, join time and temperature, the optimal design yields the largest gains with 58% of efficiency of the combined authors' proposed designs against our, and that means that by taking 42% fewer observations on our design, we would match this same precision in the joint estimate of all the parameters. So thank you so much. These are some references from our work. And yeah, that's it. Thank you so much for your attention.
Thank you very much, Alba. As I said, please, if you have any questions for Alba, please write it in the chat, and we will try to come back to those questions at the end of the session in case we have enough time. So thank you again, Alba, and we will move now to Silvia Pineda.
Hi, hello.
Hello.
Can you hear me
Yes.
and see my presentation?
Yes.
Let me share it. You see the presentation?
Yes.
Yeah.
Yes.
Okay,
Thank
perfect.
you.
So thank you very much, first of all, for the organization and the BioStatNet for the opportunity. So today I'm going to present a little bit of what we do in our group at the Universidad Complutense de Madrid. And I title it From Genes to Clinical Decisions, the Machine Learning Pathway. Here with the idea behind this is to show that machine learning techniques are important, but we need to know which is the best way to use them to extract the relevant information to take clinical decisions. So let's start by explaining you, or by saying you, that we are living in the era of the biomedical big data, especially in the omics data. The normal, or the more used to omics, the omics data that we are most used to are genomics, transcriptomics, etc. But we also have microbiome or immuno that we are working nowadays. Although today exposure factors and clinical records are also big data in this context. Nevertheless, I'm going to focus on the omics data part for today. So let's start with knowing what are omics data. So omics data come from large-scale and systematic studied molecules in living organisms, and they represent a set of technologies that enable the molecular level analysis. The components that make up biological systems are very complex, and they include interactions and relationships that occur among them and with the external environment. So all this approach have contributed greatly to the study of many of these complex diseases, such as, for example, cancer. But we are interested in analyzing this type of data methodologically speaking. So we are talking about the double challenge of omics data, because omics data has a biological complexity that I'm not going to go into detail, but we all know that this happens. But the special focus here is that they have a high dimensionality structure. And with high dimensionality, I refer that is not, or when we are talking about big data in this scenario, we are not talking about having a massive amount of observations. What we have here is a massive amount of variables. And this is our problem, that sometimes this massive amount of variables make us to have a problem where we have a number of variables much higher than our sample size. So this imbalance complicates the statistical inference, the model training, and the future selections. And this requires dimensionality reductions and regularization techniques. The challenge that we focus, or we want to focus in when we talk about high dimensional omics data, are overfitting, are dimensionality, that if we don't treat in the best way, we can lose a lot of information. We talk about computational complexity. We need powerful and fast computing resources. We talk about validation. Sometimes it's difficult because on one hand, it's hard to find independent data set, but on the other hand, the data can be very complex to really try to assess validation in a sample specific way, and sometimes are very difficult to interpret. So to deal with this challenge, we need machine learning techniques that can address many of them. However, we must avoid the black box idea of the machine learning techniques. So I'm going to show you a couple of applications that are developed by two of my PhD students, that both of them are in development, they recently joined the group, and that are focused on how to treat the data in the way that machine learnings are not this black box idea of applying it. One is how to treat data in a way that we pre-process the data before entering a model, and the other one is in the other side, how we extract the information from machine learning techniques. Both of them are an application of this next biological problem that I'm going to show briefly. So the problem we want to solve here is to differentiate between types of kidney transplant rejection using gene expression in peripheral blood. The idea here is that how to avoid an invasive technique as is a biopsy, which is necessary nowadays to really find whether an individual who has a kidney transplant is rejecting or is not rejecting the organ. So the idea is that just with a sample test, we are able to determine whether this individual is rejecting or not. So the statistical problem here is a multinomial classification and a future selection problem. So we have three clinical outcomes because we have two types of rejection, which one is antibody mediated and the other is T-cell mediated. The only thing we need to know is that these two probably have different biological pathways and an unstable one. And what we want here is to have a list of genes that they are sufficient to predict whether there is a rejection or not, but is small enough to really be able to do it in a simple blood test. So for that, we need interpretable models. So we have RNA sequencing, and we have two cohorts, one that we will use as a discovery, as a training cohort, and another as a validation. The first one has 97 patients with the 3 clinical endpoints, and the second one 37 with the other 3 clinical outcomes. The first thing we perform is applying an elastic net instead of using classical methods, with the idea that these machine learning techniques can deal with a multivariate structure, has a variable selection, and apparently are suitable for a P higher than in N scenario. We obtain a gene signature, which more or less give us 3 big clusters of the 2 types of rejection and the stables, with an AUC, an area under the curve, quite good for being a completely independent test as 0.71. Especially good when we want to discriminate one of the type of rejections against the stable, which is the 0.91. But here we observe the big problem we have, and is that when we want to discriminate between the other type of rejection, we were not able to do it. So then we thought that maybe treating the data before entering the machine learning technique could help us in improving this. So the goal here was how to pass from a P higher than N scenario to an N higher than P scenario, because it's true that machine learning techniques can deal with this problem, but indeed what we are seeing in our results is that it's not that easy. So to achieve that, we first propose reduce the number of parameters, so by filtering, in this case, very easily, univariate models with a p-value, and once we have reduced our parameters, we are going to increase our sample size using synthetic data. In this case, we are using SMOTE. Surprisingly for us was that with just reducing and filtering the number of parameters, we were able to increase a little bit the area under the curve. Not in the one that we had before, the 0.91, but we were doing a little bit better in the other two discriminations, pairwise discriminations. So we thought that maybe this is a good way to do it. So let's try what happened when we increase our sample size. So we apply SMOTE once we have reduced our parameters, multiplying by 3 our sample size. And when we observe very, very nicely is that we obtain a much higher AUC, 0.8 globally, maintaining the 0.91 that we already had discriminating the rejection group that we were discriminated before in a good way, but increasing especially the 2 other pairwise AUC that we were not able before to discriminate it. And what we observe is that this happened just in the moment where our sample size was a little bit higher than our number of parameters entering in the model. So we conclude here that by shifting to an N higher than P scenario through variable selection and synthetic data, we can increase the predictive power of the model. Secondly, we thought that we were interested in another idea that we wanted to explore because, well, the gene expression data is very complex, but we don't want to miss the possible interactions between the genes that we know that exist. So we thought that maybe trying another machine learning techniques could help. In this case, we use random forest, which is a tree-based ensemble that performs very well on complex data with future interactions. But random forest has a big problem, that is a lack of interpretability. So at the end, you have a big, massive number of trees that are not possible to integrate in the clinical applications that we are interested. So we thought that maybe treating, once we apply the random forest, all these rules that are generated by the association rules, we maybe could extract the most important ones. So we apply this idea of the association rules, which is an idea of based on the basket analysis. Basically,
yeah, sorry, we only have 1 minute left. So
okay, I'm almost done.
Okay, thank you.
So, well, basically with this idea of random forest, what we found is that we were able to go to an AUC of 0.82. And the best part of this thing is that we are able to give a rule based on our genes to establish whether we have one of the outcomes of the other. Just finishing. So basically, the idea here, or I don't need to convince anybody that we need from machine learning techniques to study information from this massive high-dimensional data, but we need to move away from using them as black box. So we should support this model by providing well-prepared inputs, selecting the most informative variables and representative samples, and we need also to make sure what is leaving the model, the outputs of the model, to really make clinical applications and understandable decisions. So the take-home message here is that machine learning is essential to unlock insights from complex high-dimensional biomedical data, and better inputs and clear outputs will transform algorithms into real clinical tools. So I jump just to the acknowledgement. So thank you very much for to the Biostatistics, and here you can find what we do in our group at the Complutense, and of course this work is done by all the students that are listed here, and nothing will be possible without them. So thank you so much.
Thank you very much, Silvia. Thank you very much to all the speakers. Unfortunately, we don't have any time left for any questions, so we have to move and let the next session start. So thank you very much, everyone. Thank you for the organizers of the conference. Thank you, Jaione, for organizing this session. Bye-bye.
Thank you all. Bye. See you.